Hello class, welcome to my presentation about the Giardia lamblia and the Orthohantavirus. I hope you'll enjoy my presentation and let's get started. So let's start with the Giardia lamblia. So it is of the kingdom Protozoa and it's a parasite that is found in the intestines of animals and humans. And it's a cyst-forming parasite, which we'll talk about it later, but it's quite important for how you actually get infected with this parasite. So when we look at the tropozoites, the first thing that is that really stands out is that it has two nuclei. And these two nuclei, they contain chromes of equal size who are both transcriptionally active. So when the tropozoites replicate, or when cell division occurs, they replicate simultaneously, so together. The other thing that you can see is the median bodies and the oxocell, and they're both part of the cytoskeleton, so they give the tropozoites stability or texture. And the other thing, it has 8 flagella for mobility. It has periapical vesicles, which take up extracellular nutrients and expel waste to outside of the cell. And this is actually, these vesicles are actually pretty important for this protozoan because it doesn't have a mitochondria. So this kind of helps the protozoan to get all its nutrition and get rid of its waste. Another thing that I forgot to mention is the ventral disc, and that's located right here beneath the nuclei, you could say. And what it does, it helps the protozoan or the parasite to attach to the inner lining of the small intestines, and that's how it gets its nutritions. So it takes up the nutrition from your intestinal cells, actually. So let's move on to the cyst. Now, this is what the cyst of the Giardia lamblia looks like. And it is actually extremely resilient. It can resist a lot of water treatment methods that we use, like chlorination and ozonation. It can survive for months in cold water, and I've read some research as well that suggests that it can actually withstand pasteurization. So you can kind of imagine what the complications are of that, and especially, because infection occurs when you ingest the cysts. So the fact that it's so hard to destroy the cysts is not really good news for us. Okay, so let's look at the structure of the cyst. The structure of the cyst, it actually has 4 nuclei. And when the cysts, or when excistation occurs, it actually hatches 2 tropozoites. So that's pretty interesting. It has a smooth outer layer and a double membrane inside here, which you can see. And yeah, I think that's what I had to say about the cyst for now. So let's move on to the next slide. The replication cycle. So how do you actually get infected, and how does this parasite replicate within the intestinal system? So you get infected through drinking water that is contaminated with feces of animals or humans that are carrying this protozoan. And the tropozoite actually does not survive in the feces, but the feces do contain the cysts, and then they end up on surfaces or on food or in water. And then when you drink it, you get infected with them. It's pretty easy to, you don't need a lot of cysts to get infected. You only need about 10 cysts for the infection to develop within your body. And well, this picture shows it pretty well. What happens is when you accidentally ingest cysts, they travel through your stomach, through your intestines, through your stomach, into your small intestines. And here the excistation occurs. So here the tropozoites form. And then what they do is they use their ventral discs to attach to the inner linings, and then they multiply. And then they multiple. Okay, I cannot pronounce that word right now. I'm so nervous. And then they multiply. And you know, they multiply in the small intestines, and then they travel through the intestinal system. And when they start reaching the big intestines, it has a lot of cysts in it. It also contains some tropozoites, but as I mentioned earlier, the tropozoites don't survive in the feces. And then the cysts travel through your intestine, through your big intestines, and they end up in your feces. And there you can contaminate yourself again if you don't wash your hands very well after going to the restroom or anything like that. Or for instance, when babies carry this parasite and you clean diapers, you can, you know, infect yourself, or you can accidentally get it on food or fomites and infect others. Oh, and I just wanted to make a small note about reproduction that occurs. So for now, it is assumed that asexual reproduction occurs through binary fission. But there's a lot of research in the subject, and that actually suggests that sexual production also might happen within our bodies. But you know, researchers are still looking into this. Okay, let's move on. Let's look at the symptoms of giardiasis, which is the sickness that is that occurs when you have a Giardia lamblia infection. It is one of the most common causes of waterborne disease in the United States, and especially in third world countries where they don't have a lot of access to clean drinking water and stuff like that. This infection is like really present. It happens a lot, and there, because the you know, they might have less access to medical help. It can cause dehydration, and there it can actually be quite deadly. But here in America, it actually almost never happens that people die of this disease. So let's look at the symptoms. It causes watery, sometimes full-smelling diarrhea. That can alternate with soft, greasy stools. It can cause fatigue, abdominal cramps and bloating. So kind of that bloated feeling you sometimes, you know, feel. It can produce a lot of gas, nausea and weight loss. And normally it might last 2 to 6 weeks, but in some people it can last longer. Or the symptoms can actually reoccur. And, you know, when we kind of talk about how your immune system, or, you know, when we kind of talk about the immune system and we look at the symptoms, you know, for instance, the production of diarrhea is one of your body's mechanisms to fight the infection, because it's trying to flush out the protozoans and the cysts. Another thing that your body kind of does to fight this parasitic infection is, for instance, making the IgA antibodies, producing the IgA antibodies, as we have learned in the previous chapters, that's an antibody that specifically fights parasites. All right. So the treatments. Metronidazole flagyl is one of the most commonly used antibiotic treatments for a Giardia infection. And the side effects that it can cause is nausea, anorexia, which actually means a lack of appetite. I know that, you know, it is associated with eating disorder as well, but in this case it means, you know, that you don't feel like eating. And vomiting. It can give a metallic or bitter taste in the mouth. It can cause headaches and dizziness. It can cause ataxia, which is actually, it kind of has an influence on the nervous system, which can like give muscle contractions and makes the coordination of your muscles and stuff more difficult. And when you take this antibiotic, it's really important not to drink any alcohol with it. Because when you drink alcohol with it, it gives disulfiram-like intolerance reaction, which means that it gives really unpleasant side effects to alcohol usage. And other treatments that are commonly used are tindiazole and nitazoxanide. Tindiazole it lays in the same group of medication as metronidazole, and it just works as well. Sometimes they give it as a combination if the Giardia develops resistance against the drug. And the nice thing about tindiazole is that it can be given as a single dose, and metronidazole, you actually have to take it for several days. I think between 5 to 10 days, kind of depending on the infection. And nitazoxanide is often prescribed to children because it comes in a liquid form, so it's easier for them to swallow. And these drugs target the DNA of the Giardia, so it causes DNA damage. And then, of course, you know, when the protozoan has enough damage in its DNA, the cell will die. So that's basically how it works. Okay, so let's talk about the hantavirus. Here you can see a picture of how it actually looks. It's pretty cool, I thought. All right, so let's talk about the structural features of the hantavirus. So the Orthohantavirus is a single layered envelope. And when we look at the, well, as you can see here, and it has two spike proteins, G1 and G2, or the GC glycoprotein and the GN glycoprotein. And the genome consists of 3 segments of single-stranded negative sense RNA, which are referred to as the L segment, the S segment, and the M segment, which basically just refers to their size. So L is the largest, M is the medium one, and S is the smallest. And the small RNA segment encodes for the nucleocapsid N protein. And the nucleocapsid N protein, what it does, it kind of attaches itself to the RNA, or it forms like a cylindrical shape around all the RNA segments, and that's what protects it. So often in class we have seen kind of this structure that lays within the envelope. This virus doesn't have that. It has like a protective shelf around every segment of its negative sense RNA. And then the medium segment encodes for the envelope spike proteins, so for G1 and G2. And then the large segment encodes for viral transcriptase and replicase of protein L. Okay, I think that's it for the structure of the virus. So let's move on. Okay, the Orthohantavirus replication cycle. When we look at the replication cycle, what happens is the viral enters the cell through endocytosis, and then in the cytoplasm it encodes itself, and then it can basically, well, then it starts transcription within the cytoplasm because it's a negative sense RNA, so it needs to copy itself first before it can start doing anything else. So that happens here in the cytoplasm. And then in the cytoplasm, it starts replicating its RNA. And what also happens is that it kind of travels through here, through the ER and the Golgi complex, where it does translation and it starts building on all the new facets it needs to make more virons. And then it assembles here at the, or it assembles here at the edge of the cell, and it puts its spike proteins on our cell wall or membrane, and then it's exited through bodying. So that's the replication cycle of the Orthohantavirus. Okay, so Orthohantavirus causes polymerase syndrome, or HPS. And you can get this syndrome when you breathe in air that's been infected with the virus or virons. And how these virons get into the air is actually through the droppings of rodents. So for instance, when you have mice in your house and they're infected with Orthohantavirus, and they pee and poop, then this virus gets airborne and it gets into the air, and once you breathe this in, it settles in the lungs and you can get HPS. So it kind of develops in 2 stages, and the first stage really looks like the common flu, which you can see it gives fevers and chills, headaches and muscle aches, vomiting, diarrhea, or abdominal pain. And then after about a week to 10 days, stage 2 starts happening, which is a cough that produces secretions, shortness of breath, fluid starts accumulating in your lungs, which is very, very unpleasant. Your blood pressure drops, and eventually it actually can influence how well your heart works. So it gives a lot of trouble in the second stage. You know, people have a lot of buildup in their lungs, they have fluid in their lungs, they get trouble breathing, and the heart gets trouble with pumping the blood around. So you could see how this, you know, could be deadly. And when we kind of talk about what does your body do to fight this syndrome, well, fever, of course, is one of the things that our body generates. And vomiting, diarrhea, and abdominal pain are often also caused by reactions of the immune system to the virus. So that are a few examples. Okay, treatment of the Orthohantavirus. So not so good news. Currently, there's no existing treatment for HBS. And that's also why it has such a, or relatively in the United States, why it has such a high mortality rate of 30%. It's because basically they cannot give you a medicine. Now, they do say, I don't know if you've ever had the flu and went to the doctor, if it was kind of, you know, long lasting, and they asked you if you had been in contact with any rodents. Why they ask that question is basically what they need to do is get you to the intensive care unit as soon as possible if you are infected with this virus. And there, for instance, they can give you, they can put you on the breathalyzer machine to help and keep your oxygen up and stuff like that, which they're treating the symptoms and trying to give your body time to fight off the virus. There are several clinical trials going on with vaccines for this virus. There are different clinical trials on DNA vaccines. There are clinical trials on activated vaccines, and I think in Korea they actually do vaccinate with inactivated vaccines for the hantavirus. But it's not been approved in the United States or Europe, so we don't use this treatment. And then there's no medical trials yet, but there's research being done on possible subunit vaccines. Okay, and then we get to the conclusion. So in really short, the Giardia lamblia is a unicellular facultative anaerobe protozoan that can be found in the small intestines as tropozoites and cysts, and it causes giardias, which is commonly treated with metronidazole. That's probably not how you say it in English, but I'm rolling with it. And then the Orthohantavirus is an enveloped virus containing 3 segments of negative sense RNA. The hantavirus is spread by rodents, and there are no approved treatments available for HBS in the USA. Sources. And then if you guys have any questions, please post them under my presentation, and I'll be very happy to answer them for you.